Hello, welcome to part 2 of our immune system lecture series and our conversations about COVID-19. For part 2, we are going to be talking about how your adaptive immune system works. So the part we're going to talk about today is antigens, antibodies, and how your immune system starts to gain memory and what that means about how vaccines work. And we'll also be talking about these guys down here in this comic, these antigen presenting cells, and how they help your body to start to gain specificity in attacking specific microbes, and how that also helps your body gain some memory. Here's the outline from your book. Last time we were together, we talked about your innate defenses, surface barriers, and internal defenses. And today we're going to start talking about adaptive defenses. In order to do that, we need to start talking about what's an antigen versus an antibody, which you should be somewhat familiar with from your labs. And we'll talk about humoral immunity and B cells. So based on your lab experiences, let's see how you do with this questions about antigens and antibodies. So which of the following is true? I'll let you read it for yourself. All right, still needs more time. Press pause. Let's walk through this together. What did you say? I can't hear you. Okay, let's walk through it. So antigens are only made by microbes. That is not true. All of your self cells have antigens that basically say, hey, I am self. The entire thing your immune system is trying to figure out is which cells in your body are part of you and should be able to stick around, and which cells in your body are not part of you, like bacteria, or not cells, like viruses that are entities that can cause disease but aren't necessarily a cell, but certainly not part of self and something that needs to get destroyed. Or it's also trying to discern if it's a self cell that's been invaded like a virus and needs to be destroyed, or if it's a self cell and something's going wrong. For instance, if it's got cancer and the immune system can target and kill those cancer cells before they grow into a larger tumor. So antigens, in addition to being made by microbes, are also made by all of our cells and are key to helping your immune system know what to keep and what to get rid of, what is self and what is other. So B, immune cells produce antibodies which bind to antigens. That is true. So that's what's producing your antibodies, are immune cells. Specifically, what we'll talk about today is you have cells called a group of white blood cells called lymphocytes, your T cells and your B cells, and it's your B cells that are producing those antibodies. And that's nice, they both have Bs. B cells produce antibodies. So that's helpful to remember. So B is the correct answer, but let's continue. We're having so much fun. C, antigens are molecules produced by the body to promote the inflammatory response. That is not true. Antigens are just produced by the body to help your immune system determine what is self and what is not self cells. Molecules that do promote the inflammatory response, there are various kinds, but one family that we've started to learn about are the cytokines. And those, there are some cytokines that promote the inflammatory response, and there's another group of cytokines that basically end the inflammatory response. But when we talk about those cytokine storms that Lorene talked about during the COVID conversation lecture, those are those molecules that are helping to promote that inflammatory response to the point where it causes damage to self cells, because it's the inflammatory immune response out of control. D, in the body, the only cells with antigens are red blood cells. That is not true. All of your body cells have antigens. So let's talk about the structure of antigens. Antigens are usually large molecules, and they're usually complex molecules and have lots of different parts to their three-dimensional structure. And each one of those parts to that three-dimensional structure can act as a binding site for an antibody. So in this case, our antigen here, you'll notice each one of these shapes is representing a different possible binding site, and that each shape has a unique interface with an antibody. So each one of these are called antigenic determinants, which is basically just a binding site for an antibody on an antigen. And each different type of determinant would have a unique antibody. So you can have multiple antibodies recognizing the same antigen. So to understand more about antigens and how they talk to antibodies as well as your immune cells, we need to talk a little bit more about how antigens get on cells in the first place. And one of the major ways this happens is through these Mhc proteins, which you learned a bit about in your Mastering A&P, but we're going to go over those again. So MHC stands for Major Histocompatibility Complex, and there are two big classes of MHC proteins, the MHC1 molecule and the MHC2 molecules, and they have some general similarities and then a couple of critical differences. And the general similarity is this: you can see that it's a protein that has a portion, one or two components that are integrated into the cell's membrane. So you should recognize these as phospholipids, although if you look really closely, I just realized those phospholipids only have one tail, and we all know they should have two. So tisk tisk on that artistry. But more importantly, you'll notice these MHC proteins have this U shape to them, and in the middle of that U shape sits this little kind of misformed Cheeto, and what that misformed Cheeto is representing in this case is an antigen. So let's watch this video and remind yourselves, if you can't quite remember from Mastering A&P, what the differences are between an MHC1 molecule and an MHC2 molecule in regards to the type of antigens that they're going to present on the outside of the cell.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
Okay, so some key differences that I hope you notice there is if you have a virus, let's say, I don't know, COVID, that has gotten into your cells, then what kind of receptor is going to respond, is going to be involved in presenting the antigens of that virus? Is it going to be MHC1 or MHC2? Yeah, MHC1. So the MHC1 receptors are only going to display proteins that actually get made inside of the cell. And as you'll recall from our last lecture, viruses actually invade the cell and use the mechanisms inside of the cell to make more viruses, which includes proteins that are on the outside coat of that virus. And so when a cell is infected with a virus, it's going to, just as a matter of course, these MHC proteins are constantly being made, bound to random proteins inside of the cell, and then displayed on the outside of the cell. So in a healthy cell, the only thing that an MHC complex is going to display are the healthy, are the self proteins as this whole process takes place. However, if our cell is infected by a virus, now those MHC class 1 molecules, instead of just presenting self antigens to say, hey, this is a self cell, now they're displaying a foreign antigen or a viral protein that flags that cell and allows those T cells, which we'll be talking about more, but T cells are another type of lymphocyte that there are some specific T cells called cytotoxin T cells that actually their job is to kill cells that have been invaded with things like a virus. Versus this, right, our phagocyte, our macrophage, which rather than asking to be killed themselves, what they're doing with their MHC2 class proteins is they're basically a billboard. They are displaying these antigens of a pathogen that they picked up, saying, hey, we're being invaded by this pathogen. Here's what it looks like. Here's the wanted poster, basically, of what this pathogen looks like. And when you find it, you should kill it. But don't kill me, kill the pathogen. And so what that MHC2 protein helps to do is that MHC2 protein saying, okay, this is a phagocyte, this is one of our own, don't kill the phagocyte, but do kill something that's displaying this pathogenic antigen. So based on what you just learned, can you answer this question?
Honey?
Hold on. Just a second, let me pause this. How'd you do? I'm back. Sorry for that little interruption. Let's walk through it. For fun, let's start at the bottom. So D, MHC1 proteins are only found on macrophages. Not true. MH1C proteins are actually found on every one of your nucleated cells. By nucleated cells, we mean cells with a nucleus. So you'll remember which cells don't have a nucleus. You're making, ooh, wait a minute. I mean a second, 1,000. You just made 2 million of them. That's your red blood cells. So your red blood cells dump out their nucleus. So those don't have MHC1 proteins. Basically everything else does, which helps for them to identify themselves as a self cell, part of us, don't kill us. C, MHC proteins catch proteins floating in the bloodstream. No, right? MHC proteins get bound to their antigens when they're inside of the cell, so they're not catching things that are just floating around in the blood plasma. If there is something in the blood plasma that becomes bound to an MHC protein, it's because that has been pulled into a cell through phagocytosis and dissolved, digested, and then a fragment of that becomes an antigen that's bound to an MHC2 receptor. B, MHC2 proteins are used to identify self cells. Oh no, my friends, you know that is MHC1 proteins are used to identify self cells. A, MHC1 proteins present proteins made within the cell. Yes, and those can either be proteins that the self would make normally to show that, hey, this is one of us, keep us around, or it could be the proteins are made from a virus and it helps the body to recognize, or your immune system to recognize that, hey, this is a cell that's been invaded by a virus. Those also help with identifying cancerous cells. That when a cell becomes cancerous, it displays some different protein antigens that the immune system, those T cells can recognize and then destroy those cells. So you have different types of cells that present antigens, or kind of are showing these little antigens flags to the immune system cells. And there are two classes: the professionals, because basically this is their full-time job and career, is to display antigens. And these ones use MHC2 proteins, dendritic and macrophages, and B lymphocytes. So these are, especially the dendritic and macrophages, are very, very good at phagocytosis. So they bring in external antigens into their bodies, and could be a whole bacteria, could be parts of dead bacteria or dead viruses, or whole viruses, parts of viruses. Anyway, bring them in, lyse them like we talked about with those macrophages in part 1. Same idea, through those lysosomes, they're going to digest those and then use those MHC2 proteins to display those antigens on the cell surface. And then the second class is, I can find a really good name for them, but we're going to call them the not so professional cells. And those use MHC1 proteins. And these are all your nucleated cells. So these are mostly just your self cells. They are constantly displaying their self antigens. And then once they get infected or once they become cancerous, they're going to present those antigens so that they can get flagged for destruction by those T cells. Here's a dendritic cell. Look. Sorry for that cliffhanger. I needed a little drink. But check out the surface area to volume ratio on that cell. Hubba hubba. Probably has something to do with its function, yeah? What could that possibly be? Could it be that, 1, it probably helps with phagocytosis, and 2, imagine all the antigens you could display with all that surface area. Fantastic. Let's watch the video and then we'll walk through this picture together.
Adaptive immune system provides.
Whoa, back it up.
The adaptive immune system provides a more targeted defense. Dendritic cells are like intelligence specialists. They collect pieces of the invader at the site of the battle. Then they travel to the nearest lymph node, triggering a cascade of events. Inside the lymph node, the dendritic cell presents pieces of the invader to T cells, the immune system's communication specialist.
In turn, T cells activate B cells, training them to form antibodies, the molecular weapons that target the invader specifically. The immune system now mounts its ultimate defense. B cells release millions of antibodies, each antibody's shape tailored to identify and neutralize the enemy. The immune system deploys these tailored weapons until the infection is wiped out, ready to respond even faster the next time.
Groovy. So this is representing a macrophage. Dendrocytes, dendritic cells work the same way, that they're going to engulf a bacterium, or they can just engulf part of that bacteria. Then they're going to lyse, they're going to have those lysosomes that fuse with the vesicle that brought in the bacteria. They're going to digest it. As they digest it, there are these small pieces of proteins that then can become antigens and then displayed with those MHC2 proteins. And that is what you just saw that dendritic cell using to train those helper T cells that then are going to activate. I know it gets marvelously complicated, isn't it so fantastic? So the dendritic cell is going to then train up this helper T cell, which is then going to help to activate these B cells, and it's the B cells that then are going to release these antibodies. And the antibodies are recognizing these specific antigens so that the body can recognize the antigens on these bacteria to help the body fight and get rid of those bacteria. So this is all that story that I just told you and watched the video about is your humoral immunity. Humor meaning, we should have a joke on this slide, right? My apologies. But humor meaning bodily fluids. So that is both your blood and your lymph. And antibodies are made by your B lymphocytes, also just known as your B cells. And one of their primary jobs is to excrete antibodies into the blood or the lymph, which is your humor. And those are going to bind to the antigens of pathogens. And one of the things that those antibodies do is basically a flag that says, kill it, get rid of this thing. So from your Labster lab, I know that you worked with this a bit and looked at antibodies and their general Y shape, and that they've got 4 peptides, meaning 4 protein chains, 2 light chains and 2 heavy chains, and those things are bonded together with a disulfide bond. But the most important thing I think for us to talk about in this case is where those antigens bind. So remember that there's this constant place here that basically that doesn't really change, but then there's this variable portion here where the antigen is able to bind to the antibody, and each antibody is unique and binds to one specific antigen. And how do you get that antigen diversity is actually a pretty fascinating story. I'm not going to go too deeply into it, but it basically has to do with your genetics, that you have some genes in your body that help to produce different antigens, but we can produce so many types of antigens, more than we even have genes for. And what your genes cleverly do is they shuffle their genetic information in different sequences. And it's basically having a deck of cards that you could shuffle in endless numbers of, almost endless numbers of sequences to get slightly different antigens, so your body can get trained to bind to all kinds of antigens. Sorry, you're making lots of different types of antibodies to potentially bind to lots of different types of antigens. So what do those antibodies actually do to help your immunity? So let's look at figure 21.15 or 15 from your book, Adaptive Defenses that Aid in Humoral Immunity. So we're just going to take it from the top. That we've got these antigens in your body that eventually get recognized by your B cells, your B lymphocyte cells, and those are going to produce antibodies and release those antibodies into your humor, into your blood and your lymph. And you're releasing, a single cell is releasing thousands of antibodies in a short period of time. Lots and lots of antibodies circulating through your body when you're fighting off an infection. So what do those do? Well, one thing they can do is neutralization. What does that mean? Well, remember that in order for a virus to invade into your cell, there has to be this protein recognition, like a lock and key, in order for the cell your self cell to be fooled to pull in the virus into its cell. Well, if the antibody coats that virus, then it's going to coat all of those proteins that the virus would normally use to bind with your cells. And so that's called neutralization, because now that virus can no longer invade a cell. Agglutination is, there are different classes of antibodies, which you worked with a bit in the Labster session as well. And these are the IgMs that have 10 different spots that and the antibody can bind with antigens. And what that means is it can cause agglutination, right? These are the red blood cells. The AB antigens are bound and recognized by that IgM class of antibodies. And those really easily form agglutination, meaning there's going to clump a bunch of cells together. And then there's also precipitation, which means that if there are soluble antigens, so an antigen that isn't connected to a cell, but it's just kind of out there, this becomes This becomes important in things like if a bacteria is releasing a neurotoxin into the body, one thing an antibody can do is actually bind those neurotoxins so that instead of causing the neurons to become damaged, they basically neutralize those neurotoxins. So that's called precipitation, when it's just taking those soluble antigens out of the body, which are, as I said, really handy when you've got a bacteria producing toxins, and your body can make antibodies to neutralize those antigens. So all of those things together, whether it's neutralization or agglutination or precipitation, those all aid phagocytosis. Why? Well, because if you have a big clump of things together, it's just easier for those neutrophils and macrophages and dendritic cells to find them. And part of what they're looking for when they find these things to engulf them are these antibodies. So this bottom of the Y, the place that's more constant in the antibody, is the portion that the phagocytes are going to recognize as something that they should engulf and digest. And so the more of those that are clumped together, the easier it is for those to find them and digest them. So that's that side of this marvelous diagram. And then on the other side, we've got this fixes and activates. So last time we were together, assuming you're watching these in order, and who wouldn't, I don't know. I'm not taking a survey on that one. But you might remember that we talked about a protein called interferon that interfered with the reproduction of viruses inside of the cell. And that was part of your innate immune response. One of the proteins we didn't talk about was this complement protein. And the complement protein, its job is to help basically all of this stuff down here. Anyway, it helps, so antibodies are going to help to activate the complement protein, which then leads to making what this purple thing right here is. The complement protein helps to make these pores inside of cell walls. And if you make this pore or this hole, it's a big enough hole that things can just easily go in and out of it, and that leads to cell lysis. And the complement protein also helps to enhance phagocytosis and just the inflammatory process in general, which is then going to help really lead to the body fighting off an infection. So that's the many ways your antibodies help you to fight off an infection. So cool. Actually, we're gonna pause. I decided I wanted to show this after all. This is a video I watched earlier today that is on antibody precipitation, and it's a cool story. So we're gonna take a commercial break.
In 1925, 20 dog sled teams ran an emergency relay across Alaska to save the town of Nome. The threat? A raging diphtheria epidemic. It would be 2 more years before a vaccine against diphtheria would become available. But diphtheria antitoxin was the next best thing. In a 675-mile race against time, the rescue mission saved the town by delivering antitoxin with its key ingredient, antibodies. A vital weapon of the immune system, antibodies are designed to fit and attach to specific invading molecules. Without them, diphtheria toxin attacks nerve cells, heart cells, and kidney cells. Once attacked by these deadly molecules, the cell stops functioning and is doomed to die. By attaching to the cell's receptors, the toxin burns through cell after cell, attacking nerves that control breathing, heart rate, or kidney function. When antibodies arrive, everything changes. Instead of attaching to the cell's receptors, the toxin now binds with surrounding antibodies, blocking it from attaching to cells. But diphtheria antitoxin provides only temporary protection because its antibodies will run out. This is called passive immunity. When the immune system makes its own antibodies, it's called active immunity, which is what a vaccine creates. Through a series of steps over many days, the immune system makes the needed antibodies, now forever ready to quickly deploy its weapons and prevent a future attack, eliminating any need for heroic dogs, brave mushers, or frozen antitoxin.
Great story, huh? So I thought that might just help you get a better handle on what we mean by precipitation and what a powerful thing that can be for antibodies to help fight infection. And of course, in that case, the antibodies were not self-made; they were injected into people, but it still helped them overcome that neurotoxin. Okay, so how does this whole process get started? As we talked about, we start with these primary response of our B lymphocytes. So your body inside of your bones are making different white blood cells, and they're making white blood cells that have different antibodies on them. And once those B cells are made, they're going to migrate into your lymph system and hang out in your lymph nodes until they encounter an antigen. And often it's the work of that dendritic cell and those helper T cells that introduce these naive B lymphocytes to their first antigen. And so you'll notice that in each one of these B cells only makes one type of antibody. So even though each cell is making different types of antibodies, a single cell is only making one type of antibody. And in this case, these this cell here with the green antibody and this cell with the pale blue antibody, right, they're not being activated. And if the body never encounters one an antigen that matches with those cells, they're never going to get activated. But in this case, this antigen's activating these antibodies, and then we get into an activated B cell, and we start to proliferate, or those cells start to divide. And as those cells start to divide, they can have two different fates. One fate is to become what's called the effector cell. An effector cell is what is actually doing the fighting or in the immune system. In the case for your B cells, that means they're going to secrete lots of antibodies into the humor, right? This is our humoral immunity. So releasing antibodies into the lymph system as well as the blood. And then those antibodies can do everything that we talked about here. Awesome. These cells live for about 45 days, and then they kind of get pooped out and they die. And your body's only going to start making those cells dividing if they continue to be notified that the antigen is still around. You also start to establish these. This is the second possible fate of this proliferation, and these are the memory B cells, and they carry these antibodies around. And unlike your plasma cells, which are the effector cells for the B lymphocytes, these memory B cells live for a really long time. They can live actually the rest of your life, and they help your, this is what establishes the memory of your immune system, which is super cool. What that means is if you encounter this antigen again, it's the same virus or the same bacteria, well, now you have immunity against it, meaning that your second response, which can be years later, you're ready. You've got a plan of attack. You've got memory cells. These memory cells are like, we remember these guys. They're out. We're going to get rid of them lickety split. And so they immediately and faster and in a bigger way start making lots and lots of plasma cells. They're going to pump out lots of antibody molecules and help your immune system just wipe that out really, really fast. And so you either don't get affected by that, you don't get sick, you don't really get any symptoms from that, or symptoms you hardly even notice, just like, wow, that was kind of crap day. It might have just been your secondary response from those awesome memory B cells helping your body get rid of that before you even realize you're sick. Or your sickness is a shorter duration and not as intense. Graph time. Antibody titer. So an antibody titer, that means your antibody levels or your antibody concentration in your plasma. And these are just arbitrary units. What's better than an arbitrary unit? Against time, so days. So something to really recognize here is here's your first exposure to antigen A. So remember how we talked about that your innate immune system is pretty rapid, that whole schematic of responses happens within minutes to hours. You are responding with your innate response. This takes almost a week, 5 to 6 days or so, for your antigen antibody immune response to get trained up enough to start recognizing what that antigen is. And it takes a few days for those cells to start to divide, and then to divide in any kind of population size within your body to really do any good. Okay, so that's your first exposure. You make some antibodies, you respond to it, and then let's say the antigen kind of disperses. And then sometime later, and it could be a few weeks, it could be years later, but because you have those memory cells that can activate your immune response really fast, then boop, as you can see, right, you get a lot more antibodies produced and a lot faster. And part of what's handy about having this response taking a while at the beginning, right? Because you kind of don't want to overreact to things too quickly. So it's good that it takes a while for it to get activated to make sure that this is really a big deal. And right, if it was a really big deal once, well, then it's probably a really big deal the second time. Let's just get rid of it quickly. So that would be your secondary immune response to your antigen. So let's sort the following into active versus passive humoral immunity. And I'm pretty sure that that video is the one we just watched. Indeed, it was. It's hard to remember my own plan sometimes. But remember, watch that video with the Huskies running across Alaska to save the city of Nome. And can you remember when they were injecting people with that those antibodies? And did that video say that was passive or active immunity? So let's think those three things. So receiving a vaccine, that'd be active or passive. Exposure to an antigen from a microbe, active or passive. Injecting someone with an antibody to COVID-19, which is something that is currently being researched. They're not, as far as I know, actively doing that yet, but that is something, as you saw in that video, that's been used in the past to help people fight off infections quickly when their own immune systems can't work fast enough to get rid of something. That's been an effective mode of treating some infections. Passage of IgG antibodies through the placenta to a fetus. Remember from lobster, those are the only types that can go through the placenta. Right, so the big question here is, how are we differentiating between active versus passive humoral immunity? That's where this diagram from your book gets in handy. So active basically means this, that your body is actively making antibodies against an antigen. So you can do that naturally, meaning that you came in contact with a pathogen and your own B cells were like, hey, we recognize this antigen, let's make some antibodies. That's considered naturally. Artificial would be you get injected with dead or attenuated. Attenuated means that they're still alive, but they're weakened. And so they tend to not cause sickness, or they might make really mild, what are those called? Symptoms, but not a full-on sickness. So that would be considered artificial because you're intentionally being injected by this vaccine that has the antigens, but then your body actively makes antibodies against those antigens versus passive. So what do you see that's common between the naturally versus artificially acquired passive humoral immunity? Yeah, that the body is being injected or acquiring antibodies. So you're passively receiving antibodies instead of actively making those antibodies. So you can naturally acquire antibodies from the mother. You can get IgG antibodies through the placenta, and then you can also get antibodies from milk. And then you can also get artificially acquired antibodies, for example, what we saw in that video where people were injected with gamma globulin shots, which is another way of saying that you're getting injected with antibodies. These are short-lived because you're not making your own antibodies. You do not have memory cells. So in a passive humoral immunity, you don't have the opportunity to make memory cells for your self. And so you're not going to have a secondary immune response with passive immunity, but it might help you get through an infection. Or in the case of a baby, will get you through until the point where your own immune system is up and running and making its own antibodies. So let's sort these into artificial versus natural humoral immunity. Receiving a vaccine would be artificial as well as active. Exposure to an antigen from a microbe would be active and totally natural. Injecting someone with the antibody to COVID-19 would be passive and artificial, while passage of IgG antibodies through the placenta to a fetus would be natural and passive. So that brings us to this conversation. How do vaccines work? Let's watch a video.
Vaccines and the immune response. How vaccines work. Influenza vaccines are able to trigger an immune response by mimicking viral infection. They are usually manufactured using inactivated or killed virus particles taken from various circulating influenza strains. Inactive fractioned viral components contain the subvirion particles hemagglutinin, HA, and neuraminidase, NA. In Canada, influenza vaccines are administered via intramuscular injection. These fractioned particles containing the foreign antigens hemagglutinin and neuraminidase are released from the vaccine into the bloodstream. There, they are met with an immune response mediated by various immune cells, including macrophages, T lymphocytes or T cells, and B lymphocytes or B cells. A major mechanism of action involves macrophage phagocytosis of hemagglutinin, breaking it into smaller components. After ingestion, macrophages display the hemagglutinin antigen on their surface in combination with a specific receptor known as the major histocompatibility complex, or MHC.
Is that MHC1 or MHC2? Oh, I hope you said 2.
T cells are now able to recognize and bind foreign antigens that are associated with the MHC. Upon binding to the MHC receptor, T cells become activated and proliferate into either cytotoxic T cells, regulatory suppressor T cells, or helper T cells. Activated helper T cells express hemagglutinin receptors specific to the vaccine strand on their surface and play a major role in antibody generation and memory B cell activation. Unlike T cells, B cells are able to ingest hemagglutinin independent of the MHC. Once internalized, B cells process the hemagglutinin antigen and present it on their surface in combination with an MHC.
2.
when activated helper T cells interact with activated B cells expressing antigen Mhc receptors, they begin secreting lymphokines, which have several effects. Lymphokines trigger activated B cell proliferation, which leads to either their differentiation into memory B cells or into plasma cells. Plasma cells produce hemagglutinin antibodies specific to the strain of influenza contained in the vaccine. Memory B cells aid in future immune response when exposed to an active influenza virus. When an
All that to say, isn't it marvelously complicated, that immune system? So much fun. That what the vaccine is doing is taking advantage of the way your immune system works, in that it's exposing your B cells through that complicated system you just watched, but basically activating your B cells to make some plasma cells, but really importantly, making those memory B cells, so that if you say get your flu shot in November and then you get exposed to the flu in January, that you have these memory B cells that then when you get exposed to that flu virus is going to right, this would be your reaction when you first get the vaccine. Here's your reaction now when you actually get exposed to the virus. And with that quick antibody response, then you either have no flu symptoms or minor flu symptoms, depending on how quickly your immune system can rally. What's that dependent upon? Well, your stress levels, how well you've been sleeping, what's your nutrition like, right? Lots of factors can impact how well your immune system is going to make that response, whether it's your first or secondary response. But that's the big story with vaccines, just taking advantage of this natural sequence of events that occurs. Along with vaccination comes this role of herd immunity, which you've probably heard about, especially as we're talking more about the possibility of making a vaccination for COVID-19, which is still probably a year out based on everything that I'm reading. It takes time for those vaccinations to be developed. And in the case of a coronavirus is in the same family of viruses that causes colds and previous SARS infections. And to date, nobody has managed to make a vaccine for that group of viruses known as the coronaviruses. Not that it can't be done, it just hasn't been done yet, which makes it a little bit more of a challenge. But in order for a vaccination to be really useful within a population, what we need is something called herd immunity, which, well, let's watch.
Vaccines protect us in two ways. Firstly, they protect the individual against serious infectious diseases. In the case of measles, for example, over 99% of people who get 2 doses of the MMR vaccine will be completely protected against catching measles. But there is a second way in which vaccines protect us. If the vaccine stops you catching the disease, you can't pass it on to anyone else. This means that when enough people in the population are protected by vaccination, it effectively stops diseases from circulating at all. This is often called herd immunity, but a better name is herd protection, because it helps to protect those who are especially vulnerable to infectious diseases. This includes all newborn babies, because they are too young to be vaccinated, older people, because our immune systems become less effective as we age, people with cancer or an organ transplant because they are on treatments that seriously weaken the immune system, and people who get vaccinated but just don't make a response to the vaccine for reasons we don't yet fully understand. You will certainly know people in some of these groups. You may have them in your family now or in the future. Perhaps you are in one of these groups yourself. If everyone around these vulnerable individuals is fully vaccinated, it will help protect them against serious diseases like measles that could kill them. So even if you don't feel at risk from a disease yourself, getting vaccinated benefits others in your community and your family. Sometimes herd immunity is used as a reason not to vaccinate. It sounds reasonable. If everyone around you is vaccinated and you are protected by herd immunity, surely it can't make any difference if you don't get vaccinated yourself. The problem is that if enough people behave like this, vaccination rates will soon fall, and their diseases will start to circulate again. This is particularly true for very infectious diseases such as measles. It's also important to realize that herd immunity doesn't guarantee protection. People who don't get vaccinated tend to cluster together in communities. So if a disease outbreak does occur, it's much more likely to spread within these communities than in the population as a whole. And herd immunity doesn't work at all for some diseases like tetanus, which is caught from bacteria in the environment rather than other people. So, getting vaccinated gives us the best chance of individual protection, as well as helping to protect some of the most vulnerable people in our society.
Thanks, YouTube. So I think that video really explained herd protection really well, which is what this graphic does as well. That the more people who are vaccinated, the less a disease can circulate in a group. And that can either happen through vaccination. You can also get herd protection if everybody's been exposed to the virus and is making their own antibodies, which is another way that herd immunity or herd protection could happen with COVID-19. If enough people get exposed to it and are making the antibodies, which is one of the reasons why they're starting to do those antibody tests with COVID-19 to figure out who in the populations had COVID-19 already and who hasn't, to get a sense of what is our societal herd protection. There are lots, as you are aware, ideas about vaccines, and not everybody is equally comfortable with vaccines. And I would encourage you, there's a link about vaccine myths that I've put up in our module, and some of you had some questions about myths about vaccines. And the problem with those myths is that they can be really dangerous to our society if it prevents people from getting vaccinated. And I think, as that video demonstrated really well, puts people who are really vulnerable to those diseases at a big risk. So some of you asked this, like, I've gotten vaccinated, or somebody in my family got vaccinated when my kids got vaccinated, but I still got sick. So what's going on there? Well, as that video stated, sometimes people don't make antibodies to the when they're exposed to the antigens through vaccines, and that's not well understood. For some reason, you're not making enough memory cells, or you never made enough memory cells in the first place for that vaccine to work. Sometimes there's a mismatch between the vaccine and the microbe antigen, which can happen sometimes in flu seasons, that they're making predictions about which flu is going to be moving around the world in a particular season. And sometimes they get it right, and sometimes they don't quite get it right. And it's not the perfect match of antigen antibody. And so sometimes in that case, you can still get sick. And it could also be that maybe you get the vaccination, you get the sickness, but it's not quite as intense as it would have been if you hadn't gotten the vaccine. And that might just be the case of what's the status of your immune system. As I mentioned before, depending on your general health, are you well rested? Are you relatively stress-free? And what's your nutrition like? And all of those are going to impact the ability of your immune system to have its full immune response. Another question that came up, why is there a new flu vaccine available every year, but you only need to be vaccinated once against something like measles, or maybe you'll receive a booster as an adult with the MMR, the measles, mumps, and rubella shot? Well, even though a virus isn't a living cell, it does have genetic material surrounded by that coat. And if you have genetic material, then it can change. That's the nature of DNA or RNA, that it can mutate. So every time a virus makes a new copy of itself, or it uses, more precisely, it uses the machinery inside of your cells to make copies of itself, then anytime you make a new copy of genetic material, you can make a mistake making that new copy of genetic material. And we call those mistakes in making copies of genetic materials mutations. And anytime you have a mutation, that means that now you could have a slightly different virus. And maybe the protein that is serving as the antigen that the antibody is recognized has changed. That there was a mutation in the gene that codes for that particular protein that's serving as the antigen. And if that thing changes shape, well, now your antibody doesn't necessarily recognize that antigen. In the case of the flu, there are different classes of flus, and different flus are active in different years. But the flus also has a relatively high enough mutation rate that we need to basically develop a new vaccine for flus every year. Fortunately, they don't change too much, so scientists have figured out how to kind of keep up with the flu and make new vaccines. And what's incredible is they do a pretty good job with that. Versus something like the measles, has a lower mutation rate, or you could think of it this way, that it sometimes those proteins on the outside of the cell, even if there's a mutation in them, those viruses don't survive very well, and so they don't become part of the larger viral population. And so the antibodies can still work because the only viruses around are the ones that haven't mutated. Hope that makes sense. And there are a couple of questions about HIVs. Let's talk about that. So HIV stands for Human Immunodeficiency Virus. Here's what it does. It targets and invades those T cells. So we've heard a little bit about T cells today. We're going to talk about them even more in round 3 of our immune system lecture series. But as you saw, those T cells are important in helping to activate those B cells to make antibodies. And T cells are also important in just kind of in obliterating and getting rid of cells that are invaded with something like a virus. So here's the issue with the virus that is known as HIV and then causes AIDS, which stands for acquired immune deficiency, so syndrome. So what happens is that the virus has proteins on it that's targeting proteins that are found on the outside of T cells. And so those viruses are entering those T cells and harboring inside of those and causing them to die and taking over their machinery so they can't do their normal job of helping your immune system. So if you can imagine, if you don't have any more T cells left, then you can't activate your B cells, you can't get your immune system started, and that leaves you without an immune system, thus the acquired immune deficiency syndrome. And then you end up dying of things that normally a healthy immune system could fight off. HIV is a retrovirus, which of course means it has a mustache and funky glasses. No, what a retrovirus is, well, let's watch the video.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens?
I'm going to pause this here because we're not going to watch that video, but here's an interesting story. And one of you asked about this, like, why are some people immune to viruses and others aren't? And part of what can make you immune to a virus, meaning you never get it ever, even if you're exposed to it, the virus never manages to get into your cells. For some people, that comes down to these guys, these little wormy looking things, these receptors that a virus needs in order to invade a cell. So for some people, their receptors are different enough that the virus can't recognize this as a cell, or the cell doesn't have a conversation with this virus and is like, oh, you look like a good friend of mine, come on in, it'll be fun. They just don't have those protein receptors. That was the case that was seen with the HIV outbreak that occurred in the 80s and 90s. And that I have a good friend who is now in his 50s, I don't know, 60s or so. And he had many friends that died of HIV and had said, he's like, I am sure that I was exposed to the virus, but I never got it. And what they found as they were starting to study HIV was that some people have a different variation of this CCR5 protein so that it doesn't bind with HIV. So even if you get exposed to the virus, it never invades your cells. So that can cause some people to be totally immune to a virus if your receptors are different enough that the virus can't invade. All right, we shall proceed.
Now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase.
This is the key part. We'll finish watching this, but what makes a retrovirus is that it has RNA that gets reverse transcribed, meaning transcription is this process where normally DNA inside of a cell is made, there's a DNA, the RNA, imagine you can see me and I turn around and start over again. Okay. Inside of a cell nucleus, normally, you have a DNA full of genetic material. In order to use that genetic material, you've got to make an RNA copy. That process of making an RNA copy is called transcription. So reverse transcription is taking the RNA and going back to DNA. Okay, so here's the key thing. That process that you just saw where he said, well, let's watch it again.
Reverse transcriptase, which...
So this process where we're going from that orangey yellow thing, which is RNA, and making a DNA copy of it, it's making mistakes. That is called a mutation. And the issue with HIV is this is does such a sloppy job that it has a really high mutation rate. And the mutation rate is so high that they haven't been able to figure out how to make a vaccine for HIV, because it mutates so fast as soon as it's too slippery. You can't figure out how to make a vaccine that recognizes an antigen because the antigens are always changing. And to date, still haven't figured out. There are some great medications out there for HIV, and lots of people now can survive for decades being HIV positive, which is amazing, but still no vaccine for HIV. And that's because it has such a high mutation rate.
Characteristic of reverse transcriptase, it has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called Integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface, it becomes embedded in the cellular membrane. And at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
So, yeah, HIV, it's a wicked one. Here's the good news about COVID. It is an RNA virus, but it doesn't do this. So it's not a retrovirus in that it doesn't do this reverse transcriptase and then do this really dubious but pretty impressive move of putting its own DNA into your own genome and your own genes. This is what makes, as he noted, HIV a lifelong infection. That once you get it, you just can't clear it because it becomes part of your own genetic material in your own cells. So the only way you could possibly get rid of it is to get rid of all of your helper T cells, which is difficult to do. And then if you actually got rid of all of them, well, then, you know, right, there goes your immune system. So here's the good news. COVID-19, not a retrovirus, just a simple RNA virus that can certainly take over cells, but does not cause a lifelong infection that your body can clear. What am I doing? We're trying to get out of this. So many buttons, it's push the button song. La, 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 la. So if you're going to draw or circle as a real little review before we leave, what part of the antibody binds to antigens? You're of course looking at an antibody. So what part would you circle? If you said this region here or this region here, you would be correct, right? Those are the variable regions. These are what's unique for each type of antibody. And remember, every B lymphocyte only makes one type of antibody, but your body has the ability to make all kinds of antibodies because it can shuffle those genes. Okay, Stanford is currently conducting an antibody study to try to determine how many people have had COVID-19. As we mentioned, to try and understand what's our herd protection at this point and trying to understand the rate at which it's spreading. There's a hypothesis out there that one of the reasons why we're seeing our rate of infection and hospitalization is much lower than what's happening in New York. And one of the thoughts is that maybe COVID-19 has been here longer than we thought. So that's one of the things they're trying to figure out. So from your lab, from PhysioX, is this where they are testing people's antibodies that they have made because they've been exposed to the virus, antigens? So if you're testing the antibodies, is that a direct or indirect ELISA test? Remembering that a indirect ELISA is binding to, so that the blue here is what would actually be on the plastic bound to the little 96 well plate. The orange here is the actual sample. So here's the antibody sample, here's the antigen sample from somebody's body. And then this green thing with a yellow dot is your secondary antibody with that enzyme on it that once you expose it to the substrate is what changes the color and lets you know whether or not the sample is positive for the antibody or the antigen. So the ELISA test for the antibody would be ding, 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 indirect, right? Because we're looking at the actual antibodies rather than the antigens. So we're using, those Stanford researchers are using an indirect ELISA, similar to a pregnancy test, which was also secondary, or excuse me, an indirect ELISA. Same thing here. They do a little prick of the finger like we did with our blood tests, and then there's little, basically a little stick that within a few minutes they can tell whether or not you have are making antibodies to the virus. Now, of course, as we looked at, it takes a few days to start making antibodies. So if you were just recently exposed to the COVID-19 and your body hasn't had enough time, hasn't had 4, 5, 6 days to start making antibodies, then you might not be testing positive yet for your antibody response. Oh, I see a white tunnel at the end. We are almost done for the day, but I thought, oh, hold on. I thought you might need some levity. This is one of my favorites. It's from an animal sanctuary in Texas.
Yeah, I think Teddy Bear found some pumpkins for Halloween. What'd you find, Ted? Pick it up. Get it? Pick it up. Good boy. Oh, what is it, Teddy? Pumpkin? Can you say pumpkin? Looks good.
I, of course, hope that this is what we all hope it is, a very happy porcupine eating a pumpkin for Halloween. See you next time. Joked.
Yum, yum.
Bye, Teddy.